Hello, welcome everyone to our streaming lecture on the male reproductive system. Thank you for coming today. So many of the things that to discuss this morning hopefully will be part of the coverage for your final examinations. So this morning our task is to discuss the male reproductive system in terms of reviewing basic science concepts on the male reproductive system, particularly with respect to development of the reproductive system, the congenital malformations associated with this reproductive development. Take a look at the basic anatomy and physiological considerations when you look at the system. Secondly, we will try to discuss common diseases affecting the male reproductive system. Okay, so this is your lateral view of the male perineum. So this is a sectional view where you can see the basic gross structures. Starting from the rightmost part, you have the bladder. Of course, lying over it is your peritoneum, and near the bladder you have your prostate, and this is the capsule of the prostate gland. Of course, you also have adjacent to the bladder, you have your bony structure, your pubic symphysis, which is a landmark in your pelvis. You have your superficial layers. You have your scarpous fascia covering these small thin muscles. Then you have your urogenital diaphragm around this area. Then you have your Bach's fascia, which covers your penile structure. Then you have your Colles fascia here also. Another view of the male reproductive system. If you look at, because this is another perspective on which you see the same structure, you have your bladder and you see the two ureteral structures on the lateral side of the bladder, and of course you have here your seminal vesicle and your prostate at the base of the bladder. Then you have your vas deferens here going to your testis, your symphysis pubis. Of course, you have your inguinal canal, you have your urethra, your epididymis, your prepuce, and your glans. And of course, your scrotal structure here is here. You have your small Cowper's gland, and you have your ejaculatory duct. If you take a look at your scrotum much more carefully in an enlarged version to the right, you see that the scrotum contains several structures. You have your inner rete testis, and you have your epididymis, and to the surface you have your seminiferous tubules covered by your tunica albuginea. And of course you have your tail. This is tail of your epididymis, which leads to your vas deferens. So those are the anatomical structures that you need to take into consideration when you look at your male reproductive system. So taking a look at your male reproductive system, take a look at the reproductive development. The first thing that we need to consider is, first, how do you determine sex or gender of a person? How does one become male? How does one become female? Of course, determination of sex according to the World Health Organization is defined by both biological and physiological characteristics. that define men and women. On the other hand, when we talk about gender versus sex is differentiated by the fact that your gender is a socially constructed rule. It's a behavior and associated with several activities that your society considers appropriate for man or woman. However, when you talk about sex, which is different from gender, which is a social construction or social identity, sex can be defined by 3 criteria. You have your genetic sex, you have your gonadal sex, and your phenotypic sex. So genetic sex is basically presence or absence of a Y chromosome. So if you have a Y chromosome, then you're considered a male. If you have no Y chromosome, then you're considered a female. On the other hand, beyond your genes, there's also the presence of the testes or the ovaries. So people will also be determined, their sex will also be determined by the presence or absence of a testis or an ovaries. So if you have an ovary, you're a female. If you're a male, then you should have a testis. And lastly, the most superficial one, I think, which is the one that is more obvious when we do a physical examination, is the phenotypic sex, which is the appearance of the external genitalia. So whether or not you can obviously see a penis, a penile structure, or a vaginal structure on the person, then that would determine his or her phenotypic sex. However, we have what we call developmental stages of your sexual development. Sometimes we have what we call the early stages of development of the human beings. We have what we call your early indifferent development stage, meaning to say, at that stage, before 7 weeks of the human life, your genes is not yet activated, so that before that period, there is no development of your reproductive organs. So we cannot really determine what kind of sex will an individual have. So the SRY gene is not activated until the 7th week of embryonic development. So reproductive organs undergo an initial common stage of development beginning in the 5th week. So beginning 5th week, there is a development of the reproductive organ, and this is called your indifferent stage. So you have your several structures. For example, your genital ducts, you have your mesonephric duct, your paramesonephric duct. So your mesonephric duct is your Wolffian duct, while your paramesonephric duct is your Mullerian duct. So your primordial germ cells, or your gonads, the development of gonads depends on your primordial germ cells, which are the precursor cells to the gametes, and they migrate out of the yolk sac wall during gastrulation. Between the 4th and 6th weeks, they return to the embryo through the GI tract surrounding the peritoneum and dorsal mesentery while undergoing mitotic division. The cells then invade the gonadal ridge, which is a mass of mesodermal tissue at the back of the abdominal cavity. Then they proliferate and become embedded medial to the developing mesonephros. And then 3 primary cell types develop in the gonadal ridge, which are your mesenchymal cells, your mesotelial cells, and your primordial germ cells. the mesonephric duct or the Wolffian duct and the paramesonephric duct or the Mullerian ducts are mesodermal derivatives that form the male and female genital duct systems respectively. So for the males, what develops is your Wolffian duct. For the females, what develops is your Mullerian duct. The mesonephric ducts are derived from the intermediate mesoderm. They form a longitudinal solid cords of tissues dorsolateral to the mesonephric tubules in the thoracic regions. And during the 6th to 10th weeks of development, each duct drains urine from the mesonephros, which functions as your temporary kidney. Okay? So, the development of your external genitalia, on the other hand, will actually depend on the presence or absence of male hormone. So even if you are, let's say, genetically male and you have testes, however, even if you have a Y chromosome and even if you have a testis, if your body is not responsive or you have a absence of your male hormone, your testosterone, and your more potent form, which is the dihydrotestosterone, there will be lack of development of internal male reproductive organs and also the lack of development of your prostate, your penis, and your scrotum. So in summary, what I'm saying is that during the 5th week up to the 7th week, you have your indifferent stage. Your indifferent stage where the development of the reproductive system will not allow you to predict whether or not it will become a male or a female. It depends on the activation on the 7th week of your SRY gene, which will lead to the development of either your ovary or your testes. Because if you have a male genetic sex, you're supposed to develop your Sertoli cells and your Leydig cells in the 8th week, and then this will produce your testosterone, which will allow for the development of the mesonephric duct, and the mesonephric duct will allow for development of your vas deferens, epididymis, and seminal vesicles, which are your internal male organs, and via your 5-alpha reductase, which is the converting enzyme which makes your testosterone more potent. The more potent male hormone will affect your indifferent external genitalia and transform that into a male penis and scrotum during the 9th to 12th week. So that is why as early as your 12th week, if you have a very good ultrasound of your fetus, probably you can already determine or detect the presence or absence of a scrotum. But of course, it needs to be validated later on by subsequent investigations. So around the 4th week, 5 mesenchymal swellings covered with ectoderm from around the cloacal membrane on the genital tubercle, and then you produce your genital tubercle, 2 urogenital folds and 2 labioscotal folds, and this gradually differentiate into your external genitalia. So as I mentioned earlier, so this is how your migration of your germ cell to the gonadal ridge happens. So that they migrate from your GI tract they migrate from your GI tract towards your gonadal ridge, so that they will now form your primordial germ cells, and then they will become your primary sex cords. So this gonadal ridge is adjacent to your mesonephros, which is your early renal system, which drains the urine of the fetus. So you have a migration from the GI tract to the gonadal ridge, so that you will have normal development of your sexual organs. So what are the hormones that are in charge of sexual differentiation? You have your testosterone. Testosterone stimulates development of the vas deferens in seminal vesicles in your epididymis. On the other hand, you have your Mullerian inhibiting factor, which inhibits the development of the ovaries in the fallopian tubes. So that, because in the genetic template, when God designed us, the female sex is the default template, meaning to say, in the absence of any other stimulation, a human being will become a female. So that you need hormones like your testosterone and your Mullerian inhibiting factor to inhibit the development of the ovaries and the fallopian tubes. So that goes to say that females were the first creation of God, which basically I think controversial, but this just shows to you that the female sex is the basic template in the development of our reproductive system. You also have your dihydrotestosterone, which stimulates the development of the prostate gland. In terms of your external genitalia, the DHT, or the dihydrotestosterone, the more potent form of your testosterone, introduction of your 5-alpha reductase, also stimulates the development of the penis and the scrotum. Your estrogen, on the other hand, is needed for the development of the vagina and the labia. So, however, estrogen is not needed for the development of ovaries and fallopian tubes. In the absence of any other stimulation, anybody can develop an ovary and a fallopian tube if you don't have your male hormones, especially your Mullerian inhibiting factor. Okay, so taking a closer look at your overview of your sexual differentiation, you would see therefore that once the primordial germ cells migrate to the gonadal ridge, there is now a potential for the development of either a male or a female individual. So that through your SRY gene, you have, if in the absence of any stimulation, eventually a fetus or an embryo, it would develop into a female. So that's the default template, meaning to say that in the absence of anything, any stimulation, it will become a female. However, with the presence of your SRY gene, you develop your testes, and then your testes, due to production of your hormones, will lead to the development of your Sertoli and your Leydig cells. In your Sertoli cells will lead to the development of your Mullerian inhibiting factor, which will suppress the development of the ovary and the fallopian tube, or suppress your the development of your paramesonephric duct into uterus, fallopian tubes, and your upper vagina. So you need your MIF, or Mullerian inhibiting factor, to suppress the development of your paramesonephric duct into a female genitalia. On the other hand, your mesonephric duct is transformed by testosterone into vas deferens epididymis in your seminal vesicle. And once your testosterone is converted to dihydrotestosterone, which is the more potent 1,5-alpha reductase, your phallus, your external genitalium, becomes developed to the male external genitalia called your penis. So that is how male external genitalia develops. So your testis needs your SRY gene, which acts on your indifferent gonads. So your TDF, your testis determining factor, acts on the indifferent gonads, allow it to develop into your male gonads, and you will have the development of your primary sex cords. You have your coiled testis cords, you have your straight rete testis, and then your rete testis plus your mesonephric tubule will become your efferent ductules, while your mesenchymal thickening will be your tunica albuginea, and then you will have your mesothelial cells, Sertoli cells, and then you have your mesothelial cells between your testis cords will develop into your Leydig cells. They follow the path of the gubernaculum into the inguinal canal. So, therefore, you have the development of both your internal genitalia and your external genitalia, so that your internal genitalia, you have your vas deferens, epididymis, seminal vesicle, including your prostate, is by the action of your testosterone, while your external genitalia is your penis, scrotum, is through the action of your dihydrotestosterone, the more active form of your testosterone hormones. So, in terms of the internal genitalia development, at the 8 weeks, your Sertoli cells will lead to the development of your Muller inhibiting hormone, and it will lead to the regression of the Mullerian duct, while your Leydig cells will produce your testosterone and lead to the differentiation of your Mullerian duct into male reproductive organs. Your mesonephric duct differentiation then will lead into the development of your seminal vesicles, which is your terminal end, your epididymis, which is composed of your coiled ducts and caudal portions, ejaculatory duct at the prostatic urethra, and your ductus deferens will have your smooth muscle coat. Your prostate, on the other hand, is an endodermal outgrowth from your urogenital sinus, and the outer zone of your prostate glands are actually in the posterior lobe, while the inner zone of your mucous gland are found in the median lobe. So in terms of your external genitalia, you have your phallus enlargement, you have your development of your penis, glans penis, you also have your urogenital fusion of the body, the midline, then you have your labioscrotal fold fusion, which leads to the development of your scrotum. However, not everything goes according to the design which it was meant for, and that is why sometimes we have what we call congenital malformations, like gonadal agenesis, hermaphroditism, pseudohermaphroditism, cryptorchidism, congenital inguinal hernia, hypospadias, and also epispadias. So these malformations are actually very common among males. So let's take a look at each of these malformations and how they actually are defined. But first, before we go to that, to the specific cases, let's take a look at this particular case of a 24-year-old man being evaluated for infertility, and during physical examination, the urethral orifice is noted to be on the ventral surface of the penis. Which of the following is the basic defect that caused this abnormality? A. Abnormal development of the prepuce. B. Abnormal fusion of the paramesomorphic ducts. C. Exostrophy of the bladder. D. Failure of the urethral folds to close. E. Repeated inflammation of the glands and prepuce. So, what is the basic defect that causes this abnormality? So, you have an abnormal location for the urethral orifice through which your urine exits from the body. So instead of it being in its normal position, it's on the ventral surface of the penis. So this abnormality is caused by what defect? So you have your choices. You can take a guess at it. And while you're thinking, let's take a pause and we'll go back after a few minutes.